Antimicrobial drugs function by exploiting structural and biochemical differences between microbial cells and host cells. The most effective drugs target processes or structures essential to microbial survival that are absent or sufficiently different in the host. These selective targets include the bacterial cell wall, prokaryotic ribosomes, unique metabolic pathways, and nucleic acid synthesis enzymes. The concept of selective toxicity is central to antimicrobial chemotherapy, ensuring that drugs damage the pathogen while minimizing harm to the host. Cell wall targeting drugs such as beta lactam antibiotics include the cross linking of peptidoglycan, a critical component of bacterial cell walls. Without proper cross linking, the cell wall becomes structurally weak and unable to withstand osmotic pressure, leading to cell lysis. Because human cells lack peptidoglycan, these drugs are generally highly selective and less toxic to the host. Protein synthesis inhibitors exploit differences between prokaryotic and eukaryotic ribosomes. Bacterial ribosomes are 70S, while eukaryotic cytoplasmic ribosomes are 80S. Many antibiotics bind specifically to the 30S or 50S subunits of bacterial ribosomes, disrupting translation. However, because mitochondria possesses ribosomes resembling bacterial 70S ribosomes, some drugs can produce adverse effects in host cells. Antimetabolites, such as sulfur drugs, interfere with metabolic pathways unique to microbes. These drugs mimic essential substrates such as PABA and competitive inhibit enzymes involved in folic acid synthesis. Since humans obtain folic acid from their diet rather than synthesizing it, this pathway provides another example of selective toxicity.
Antimicrobial drug actions. Inhibit cell wall synthesis. Beta-lactams, penicillin. Prevent peptidoglycan cross-linking. Inhibit protein synthesis. Target ribosomes. 70S ribosomes in prokaryotes. Major drug classes: aminoglycosides, tetracyclines, macrolides. Inhibit nucleic acid synthesis, DNA or RNA synthesis inhibitors. Antimetabolites compete with PABA. Sulfa drugs interfere with folic acid synthesis. Drug effects: bacteriostatic, inhibit growth. Bactericidal, kill microbes.